Olá a todos e todas, eu sou Vilma Loreto, do Departamento de Genética da UFPE, e vocês estão acompanhando a mais um episódio do Podcast Transcrição. No episódio de hoje, damos continuidade à nossa série da internacionalização. Nesse episódio, teremos a participação da professora Ana Maria Benco, do Departamento de Genética, e da professora Katharina Nargar, diretamente da Austrália.
Hello, Katharina.
Hello, Anna. How are you?
Fine, and you?
Very well, thank you.
Thank you very much for participating in our podcast. It's a great honor to have you here, talking with us a little bit about your career, your trajectory. Yeah, we have about 40 minutes to talk. I think this is a lot, but you can you can stay completely well to talk something else besides what I'm asking you.
Mm-hmm.
And our first question is, how was your choice for biology? If you choose to work with living organisms when you were very young or later, how was it?
So I didn't have the clear path already very early on. It was more at high school that I really, I always loved biology, but at high school I really understood that that was great interest of mine. And I think it was the curiosity about the natural world and how complex it is, and that there are so many things that you can learn about it, but also that there are so many things that we still don't fully understand and that they are still out there to discover, that attracted me. Another thing is that I think I lean towards the scientific method as well, so that there's a methodology that you can use for scientific inquiry, and that if you follow that you will obtain results that based on the data that you gathered, and that will tell you something about the world that you may not have known before or wasn't known before. So that intrigued me. And the third one was that I was feeling that working in science would also deliver something for the greater good, that it wasn't just something for myself, but that I could contribute to something that was greater, and make a contribution to that. And that I found also really fascinating about biology and science. So that were the things that attracted me to biology.
When we met, it was in 1999 in Frankfurt, as I met you the first time. At that time, you had already chosen to work with plant biology, plant evolution. How was this choice?
That was also not quite straightforward. So when I started studying biology, I was aware that there are many fields and that I hadn't quite a good understanding of what they really entailed. And I was mainly contemplating going into ecology at the start, because that was the most complex thing, understanding the whole world and how everything is connected with each other and works together. And also the aspect of then being able maybe to help protect that really complex environment. I then was in my studies attracted to also the aesthetics of anatomy and morphology, but I didn't get the sense back then that there's so much still to discover. I thought it was more all, the basics were known and so on. And I set out then to do my master's project in Venezuela in a ecology project, vegetation ecology. in the lowland rainforests. There was a canopy crane, and my task was to map out the distribution of lianas in a three-dimensional space in different parts of the forest, disturbed, undisturbed. And it was only there that it struck me that in highly biodiverse countries, there's so much that we don't know, and even just putting a name on species was a challenge. So I came back with the sense that we are not quite there yet with doing something that complex in such a complex environment, and that studying the fundamentals would be really important. And then was offered a PhD in plant systematics. And at the time, I didn't quite yet grasp that plant systematics is really the key to evolutionary research. And by generating those frameworks, evolutionary frameworks, and also studying traits that we understand more about plant evolution, because in my undergraduate studies, I got more the sense that it was looking at fossils or be a bit more, not so much based on the contemporary biodiversity.
Mm-hmm.
So that was my transition then into plant evolutionary research.
Let me ask, who was your master advisor?
Yeah, that was Professor Moravec, and we met in that area before as well.
And you worked in a crane in Venezuela,
Yeah.
How was that?
Yeah, that was quite an adventure. It was
very
big
full
adventure.
of emotion,
Because if you are, so the crane was 30 meters tall, and it had a gondola, and we had to do a driving license for that crane because it was on a 100-meter railway track, so that we would have 1.5 hectares to work on that crane. And in my shift, I would be the only person on the crane. I would be in the gondola with the little remote control and control the crane. That was a bit scary. And the other thing is that the gondola was made out of metal. And above the canopy, of course, it's like in a desert. It's really hot. and so it was also a bit challenging from that regard. And the long crane, it also swings and sways a lot. So you almost get a bit of seasick swaying in the air. But of course, it was an amazing experience. And it was set near a table mountain, Cerro Duida. So it was a beautiful aspect of the Amazon.
I believe. I didn't know you worked with fungi before working with plants. And how was this change? And how did you choose the project for the PhD, or it was a proposal of your advisor?
So the project in the Amazon was proposed by Professor Moravec, and it was the liana diversity in the canopy. And for my PhD project, which was on bromeliads, we set out, because I liked anatomy, to do a leaf anatomical study of bromeliads for systematics. And it only then developed into a molecular study because my supervisor, Professor Zitzka, had a collaboration with Professor Karl, and just started to get into molecular systematics, which was a nascent field at the time. It was really very novel back then to do the first molecular studies for plants.
And for this bromeliad project, where did you collect samples? Did you had field trips? How it was?
Yeah, so the start of the project was that the Palm Garden, so the Botanic Garden in Frankfurt, had a huge bromeliad collection a huge diversity already there. And another botanic garden in Heidelberg also had a massive bromeliad collection. So that was the starting point for my leaf anatomical studies. And so I then started also to do field work in South and Central America for understanding the natural diversity and the habitats that bromeliads come from. My first travel to South America was already in my undergraduate, and that was because there was NGO next to our university that looked for volunteers to work on a conservation project, and I put my hands up for that, and went to Costa Rica and fell in love with the tropics and thought I would need to specialize in tropical plant diversity. So yeah. I then went to Cuba and Panama because our university had exchange programs there, and we went there as a student group to do field excursions together with students from those countries, and they would also, some of them would also come to Germany. And I did field work in Bolivia for a bromeliad project for a genus in the Andes, Fosterella, during my PhD studies. And of course, went to Brazil a few times and collected bromeliads with your group and also with Grasa Vanderley's group. That was very exciting.
And also Elton Lemme, yeah.
Yeah, Elton Lemme as well. Yeah, his collections, yeah.
And I was wondering while we were talking, some people say the young students, they don't like so much to travel as before. Do you have that feeling also with your students? I heard this also from German colleagues now. In March, I was there. Do you believe also this is a problem that people are not willing to travel abroad so much as before?
I only had one case, and that is a couple of years back, where we had trouble convincing a very promising candidate to do fieldwork in another country, and they then decided it wasn't for them, and they looked for another topic. But I find here in Australia, for example, the students are very enthusiastic to have fieldwork components and go out into the field and travel abroad. I think that's, I know I can't see that trend. And I'm very happy that there are still young people who want to see something from the world and see the plants or organisms out in the field. Yeah.
And then after doing your PhD, how it was?
Yeah, so after my PhD, I started my postdoc still based in Germany, and we had some fantastic collaborations with our South American partners and had PhD students come to our lab to do their parts of their degree in the molecular lab in Frankfurt. And then in 2010, I think it was, I was in Brazil doing some field work when there was an email with an advertisement for a position in Australia. So at the Australian Tropical Herbarium. And what attracted me to apply for that position was that it was the first time that I saw an advertisement for a molecular systematist that is based in the tropics. And it was advertised internationally, so I applied for that. And it was initially a 3-year position. So I didn't plan to forever leave Germany. But then it turned out, there was then 3 years later a permanent position becoming available, for which I applied, and now I'm here since 13 years.
13 years, a lot of time.
It
didn't look so much for me. I thought it was later. Because when we had that ProBrau project with Frankfurt, you were already in Australia. Now I remember, it's true. And going back a little bit, you have a son, and then you had your son during your PhD, I believe, yes, or was during the postdoc?
It was towards the end of my PhD, because I thought the PhD was nearly ready, but things always take a bit longer. So he was born a year before I submitted my PhD.
How it was to have a son in the middle of your studying, finishing a PhD.
So I had already at the International Botanical Congress, I think it was in Vienna, made acquaintance with Mark Chase, and he was willing to take me on for a postdoc project on bromeliads as a guest researcher. And when I then found out that I was pregnant, I decided to do that first before my son is born, because then afterwards travel would be more difficult. So I went there and worked in the lab and worked on molecular, developing a nuclear marker for bromeliads, which was really difficult at the time. And came back, I think 7 weeks before my son was born, back to Germany. Then in terms of how it was to be a young mother with a child and still finishing up my PhD and then continuing with research, I found it a bit challenging at the start, because from having 150% of your time before that you can, really give your all for your science and your project, then being reduced to maybe a 70% or sometimes less, I found that was challenging and demanding. But then I think it is a really good learning experience to be really very, very efficient with your time.
Time management, complete.
And it was a hard lesson, but I think that it was a good lesson, and it set me up for then later on, also to stay quiet, to prioritize, to know where you dedicate your time to. And the other thing, I think I started really early to then collaborate more, to work with students, train students to do components of projects, and have building a research group in a sense. So I think it had lots of benefits, but it wasn't the easy route, as you would probably agree, that you first have everything under control, and then having a bit more having a bit more dynamic in your life with a little child.
And how old is your son now?
My son just turned 17.
17,
Is
a very good product of your PhD. I had also my oldest son during my PhD, and this was really a challenge for me, especially because I was far from all family. No support from the family was possible. Did you have some family support?
I had family support, mainly my son's father, who was able to work from home. So that was a huge support.
Oh, yes. What about Australia? Did you change your research after getting to Australia, or for some time you continued working in the same projects? How it was this moving to Australia in terms of your research?
Yeah, so there was a transitional period because when I left, I had co-supervised a couple of PhD students at the time and collaborations with Brazilian students as well. So I continued to work with them on their projects and on the publications. That probably another 3 or 4 years. However, my position was to study on a significant plant group of the Australian flora. And so I had to switch organisms because bromeliads, of course, are
they don't occur in Australia
to Latin America. And so I wanted to find a plant group that was similarly in life form and also in diversity and in the challenges that it would present. And of course, very similar in that regard are orchids. So I decided to embark on another systematic plant group. And it was, of course, a big learning curve because there are also lots of them and they are complicated. And there was a lot of literature to get my head around. It felt like being, jumping into the deep end again after having familiarized over a decade. I had worked on bromeliads. And come to a new country and be not known by anybody here and start on a new group was like a new beginning. But the Australian colleagues were very, very accommodating and helping and very enthusiastic. And I found very good collaborators as well that really accelerated the speed with which I could start designing projects, getting funding, and developing a new line of research here in Australia. But there was a transition period, and that was good. I think when you start something new, there's a lag time until those projects develop and you have the publications that you can write up. So I think it is always a transition necessary when you start somewhere new. Yeah.
So you are working now with orchids. Yes?
Yeah.
Yeah. Everywhere, nice plants you choose.
Yeah.
Tell us a bit about your actual research project.
Yeah, so I have, I would say, 3 main lines of research in terms of topics. So one is plant systematics, phylogenomics, trait evolution, diversification rates, historical biogeography. So that is mainly based around molecular phylogenetics, phylogenetic frameworks, and then different molecular, morphological, physiological, ecological characters that are then investigated and then mapped and reconstructed based on those phylogenies, doing molecular clock analysis, looking at historical biogeography also based on those phylogenies. This is one area of research, and my main study group are Australian orchids now, but I also supervise students that work on other plant groups, for example, pitcher plants, or a student that finished a while ago was working on the large plant order Sapindalis. So that's flexible. Then the other line of research is at a lower taxonomic level, where I look at species delimitation in species complexes, and they intrigue me a lot. Groups that have radiated only recently, where the molecular signature is not so clear, where finer resolving markers are required. Plants that are, and they often are, that hybridize, that don't have those clear-cut boundaries, intrigue me.
These lower groups are within the Orchidaceae, yes?
Yeah,
Okay.
Within Orchidaceae as well. So these are conservation genomic studies often on plant, so species complexes of orchids that contain rare and threatened orchids. But because species delimitation in orchids, because of their plasticity plus their variation, is not so clear cut. And then there are, of course, a lot of orchid enthusiasts who also like to distinguish a lot of different species, describe a lot of new species.
Yes, and this is the difficult, because they are often not valid species, when you look in all these parameters,
And that the speed at which orchids are described is really high. And in Australia, about 20 years ago, we had around 900 species, and now we have we have about 1,600 species, so almost doubled. And for conservation, it means that there are a lot of species where it is not clear whether they are valid species and whether they need to be protected or not. And so they are not listed on, often not listed because they are data deficient, where it can't be decided whether they are proper species or not, where the species delimitations are. And so it is a really important field of study to help clarify those for informing those listings, but also then conservation action in terms of what is the genetic diversity within those groups and so on. So, so that is another field of study. And another field that is an more emerging field, but I've followed it for many years and have always been intrigued with that, is molecular identification of plants. So about 20 years ago, there were the first barcoding initiatives based on one or two markers. But it was then pretty clear early on that for plants that isn't sufficient, and that we can't discriminate species that easily. Nowadays that we have new marker approaches, that field is now very dynamic and very promising. So I'm doing some work in that area as well. And I find that very fascinating, because it would enable us to understand our diversity quicker if we had faster means of...
Maybe, in plants, the trend is to combine the different markers, especially nuclear and plastidial markers, I would like later to know more about this. Do you have publications about that?
No, but I have currently a large project that is a 3-year project where we explore the limits of that with a technology, target capture sequencing, or hybrid enrichment it's called, where with high throughput sequencing we can sequence many nuclear markers.
Nice. What about this climate modeling? Do you still do work with that? Can you explain more or less in simple words to our audience what is possible to do with this kind of approach?
So the work we've done in this area is based on climate models for the region, for example, for the whole of Australia, or I'm in a region that is called Australian Wet Tropics, which is a UNESCO listed World Heritage area with rainforest, and a very unique rainforest because it has a lot of very old lineages in there. So we have models for the current climate, and then models for the past climates, during the glacier and interglacial times about 19,000 years ago that were developed for this area. And then for the whole of Australia, also predictions, climate model predictions for the next 100 years around. And under different climate scenarios, what happens if no action is taken or some action is taken and so on. So based on these models, we can then use plant distributions today to assess the climate niche of the plant species based on the current distributions and the climate models, and then predict their distribution under changing climates, either how have those distributions been in the past or how will they be in the future. And a study that we've done on a very widespread Australian orchid, and that is an exception, usually they are not very widespread, was on the rock orchid, which is an epiphytic, so occurring either on trees or on rocks species in the Dendrobium orchid group. And so that species occurs along the Australian east coast for about 2,000 kilometers in length. And it was unclear how many species there were in that group. And the concepts range between 1 species or 11 species. And my honor student at the time did a molecular study on that and found that it is only 1 species with 2 subspecies. There was a really clear divergence in lineages. And she was a little bit disappointed at the time, thought, she should have seen more structure in this group, or that it's only 1 species or 2 subspecies. Then we did the climate modeling in the past, and it turned out that now they have a nearly continuous distribution along the east coast, but the climate model showed that when it was drier in the glacial times in Australia, there was a large gap along the east coast. And that is exactly where the 2 lineages split then into the 2 subspecies. Because of, so all the samples from the 1 subspecies was above that barrier, and all the others were further south. And yeah, that was such a great discovery, because nobody had ever known about that structure, and nor was able to explain it. And the climate modeling into the past showed that was based on this disjunct distribution that the species had in the past.
It's fascinating. Yeah. It's very nice to be able to do that. Yeah, to look in the past and then to modulate. Yeah, or to model these possibilities. Yeah, and what about the future? In the future, there are predictions of more heat and less rain. How is it in Australia? In Brazil, we are very concerned about this climate change and what is happening now, and some things that are coming faster than we expected. And then this modeling, would it help to plan for conservation or for other measurements in the future?
Yeah. So of course Australia is under similar pressures, and our Australian wet tropics region, which is a mountainous region, we have a rise, the Great Dividing Range that runs parallel to the coast. And we have the tropical rainforest here, and in its higher altitudes, it contains a lot of endemic species, and climate modeling that was done at the herbarium here showed that with changing climates, under all scenarios, even the most optimistic scenarios, there will be a significant loss in endemic species based on the shifts, because the cold adapted species which require more moisture will have nowhere to go. They shift up the mountain and then they can't continue, and then they have trouble to survive. So I have my previous PhD student did a genetic study on some epiphytic orchids that are endemic to our mountaintops here in the Bulbophyllum alliance. So it's a large genus, the second largest genus in orchids. So she did population level studies across the distribution of the wet tropics and looked at the population structure of a few of those species. And the reason was that we will probably not be able to maintain that diversity in situ. So these need to be grown somewhere else to protect them in other climates.
Have them in collections. Mm-hmm.
Yeah, and so to know
collection.
Ex situ collections, and to know from which mountains you need to collect them to have the full diversity. We did those genetic studies, and it was really interesting because we have lots of mountain peaks here, but she could identify very clearly centers where they had uniform distributions, and where you didn't need to collect from each mountaintop, but just from that region to represent the genetic diversity. And during the glacials and interglacials, there were different, in the Australian wet tropics, different centers where distributions shrunk to and then expanded from that, and that causes
these refuges.
Refuge, yeah. And so because from those different refuges, if you sample from those, then you have the genetic diversity represented. So there's now a larger project that is led by the Herbarium to sample endemics from the Australian wet tropics mountain tops and have them propagated in different botanic gardens in Australia.
Now, this is really something to be worried about, to hear of that. We have a similar problem here in this mountain wet forest, these brejos, the altitude we have. And this is really to be hurried about. What about these mega fires in Australia? Recently, we heard a lot about that. We have some also in Brazil. And is this also threatening the biodiversity, in your opinion?
Yeah, so
because there are fire-adapted flora also in Australia, isn't it?
Yeah, so the fires that we had in 2019, 2020 were unprecedented. So in the 200-year history of records, there was never such a huge fire and over such a long time and with that intensity. That was on an area of 8 million hectares. And 50% of the known plant populations from that area and their ranges were affected. So it was very, very intense. The study that I contributed to that was published about those fires found that most of the species that were growing in that area are actually adapted to summer fires. So those fires were in the summer period. The Australian vegetation has adapted to summer fires with different means. And so in a sense, it was not an unprepared community, but there were still things that were exceptional about it, and that also has to do with the changes in species in the Australian landscape, because it's not that we only have native species here. We have now a lot of species that were brought to Australia that changed the dynamics of those fires and lets them burn hotter. There are many grasses that were introduced that produce a lot of fuel load that native grasses hadn't produced. And that meant that the forests burn hotter and that the fires reach further into the canopies. The sub results or components of the study that I contributed to the orchid part with my colleague was about looking at the effects on orchids, and epiphytic orchids were most severely affected because they are not adapted to those really hot fires, and they don't have a seed bank in the soil. And so in the natural environment, they would not have been exposed to those conditions. Whereas the Australian terrestrial orchids, the majority of them hibernates in summer because of the drought and because of the regular fires. They have their geophytes, so they have underground tubers, and they are then underground, and heat and fire can also stimulate them to then grow in the season after fire. Also the nutrients that are then released and into the soil, and the opening up of the vegetation is beneficial for those ground orchids. There were only few or relatively few orchids that don't have those mechanisms that are ground orchids, and they of course then also suffered.
And finally, I would like to know if you have plans to collaborate still or new collaboration with South America.
So I have quite a lot of collaborations nationally and internationally. I regularly have students that come here to do their master's degree. I just had a German student that completed her master's. I will get another German student, and that's just a coincidence.
It's not from your prior group of work. It's another group that sent the students.
Yes, so one student got stranded here coincidentally during COVID, and then contacted me and just wanted to know whether she could do some voluntary work because she couldn't go back to Germany. And then she started doing her master's project with me here and completed that. With South America, I currently have no student coming, but I would always be happy to host them. I have had Brazilian students before, and they're always very eager to learn and very good students. I've really loved that interchange, and knowing then
Let's provide some students.
And also knowing that they go back into their countries with that new knowledge and can again teach other students and
this
propagate
is very important. Thank you so much, Katharina. It was so nice to see you again, to talk with you. I believe our students, our colleagues, they will be very interested in your research. They will look for and read more about what you are doing. We have also a lot of our kids, so maybe this is also a possibility. A lot of people working on orchids taxonomy and biosystematics, not so many phylogeny, so maybe this is really a good possibility to interact to Brazilian groups and to receive also students from Brazil. We have now a new government, and I believe this will also bring us new financing possibilities. Let's talk about in the future.
Absolutely.
Thank you so much.
Thank you so much for inviting me. It was really a pleasure talking with you.
Então, este nosso episódio, ele teve o apoio do Programa de Pós-Graduação em Genética e Biologia Molecular, do Departamento de Genética, e do DRI, a Diretoria de Relações Internacionais da Universidade Federal de Pernambuco. E vocês já sabem, podem continuar nos acompanhando no nosso canal do YouTube e também no seu agregador de podcast favorito. E até o próximo episódio, pessoal.